This video is about the recent publication Birth of the Eukaryotes by a set of reactive innovations, new insights, forces to relinquish gradual models. So it's about how eukaryotes came to be. And my name is Dave Speijer. I work at the Faculty of Medical Biochemistry at the AMC in Amsterdam, the Netherlands. And actually this work was triggered by a publication by Elizabeth Pennisi in Science of November last year, entitled Modern Symbionts Inside Cells Mimic Organelle Evolution. And what actually was implied in the interviews that were described in this in-depth article in Science was what some of the participants of a colloquium meeting in California about the evolution of organelles by endosymbiosis seemed to imply. And what they implied was that actually endosymbiosis is no big deal. It's quite easy to obtain, and we have modern examples. And the other implication was that this holds true for all forms of endosymbiosis, even the most important, crucial one, in which an alpha proteobacterium was taken up by a host cell, and this gave rise to mitochondria. And actually, I completely disagree. I think that this first endosymbiosis, this first uptake, really gave rise to a lot of quick and revolutionary adaptations inside the host cell, and that all these adaptations make all later acquisitions much more easy. So that's really a different model. So what are the two competing models when it comes to the rise of the eukaryotes? And by the way, the rise of the eukaryotes was rather late in evolution. And we have bacteria and archaea appearing on the scene almost instantaneously after temperatures in the beginning Earth dropped. And then we have to wait 1.5 billion years again for the sudden appearance of eukaryotes. Why was this so sudden? Why did it take such a long time? Because I think this reflects an evolutionary bottleneck, and that evolutionary bottleneck is the acquisition of the first endosymbiont. This really was a merger and not a simple acquisition of a relatively small organism inside already a big eukaryote. So what are the two competing models? The two competing models are symbiogenesis on the one hand, and that really is the idea that an archaea, still a complete archaea, not a pre-eukaryote, takes up an alpha proteobacterium, and that this gives rise to the full-fledged eukaryotes that we observe today. And the alternative theory, which I don't agree with, is the archezoan model, in which we have tiny, small, gradual steps towards a complete eukaryote, and that as its crowning achievement, this pre-eukaryote takes up an alpha proteobacterium and gets a mitochondrion, and then we have the eukaryotes as we observe them today. It's clear that there is a major problem with this archezoan model. If we look at modern eukaryotes, modern present-day eukaryotes, they need an enormous amount of ATP, they need an enormous amount of energy, because they are much more complicated, they are bigger, they have much more proofreading capacities in their polymerases, their protein synthesis is more reliable, they have more post-translational modifications, much higher levels of complexity. And that means you need the ATP. And this ATP comes in enormous abundances from the linking together of the different metabolic routes that the host, the archaea, and the endosymbiont, the alpha proteobacterium, brought into the merger. So we have an integration of metabolic pathways, and we have a very efficient process, the mitochondrial respiratory chain, that generates an enormous amount of ATP, and that's needed for the development of all the possibilities of the eukaryote. But you always pay a price in evolution, and the price you pay here is that that final acceptor also leads to reactive oxygen species. And one of my personal favorite models is that not only do we now have internal reactive oxygen species and not on the outside of the alpha proteobacterium. We have them inside the cell. We also make more, and that has to do with the integration of different metabolic pathways with different acceptors and intermediates like FADH2 and NADH, electron carriers. And the simple fact that glucose has a different kind of ratio of these two carriers than, for instance, fatty acid breakdown. This, I think, gave rise to the peroxisomes. And the interesting thing is that I predicted peroxisomes to be later adaptations of the eukaryotic cell and not present before the arrival of the mitochondria. And this has now been borne out by recent research published also in BioEssays by Bolte et al. By the way, if you look at recent discoveries, they all seem to really prove that symbiogenesis is the correct theory. Not only phylogenomics shows the eukaryotes to be firmly associated with a certain class of the archaea, but that class of archaea now seems to have possibilities to endocytose already, and this is one of the eukaryotic features that really already was present in the simple archaea, to endocytose other bacteria, for instance, as a source of food. In the area of phylogenomics, I should really mention the work of both Embley and colleagues and Martin and colleagues that really show that symbiogenesis is the correct theory and that the archezoan hypothesis really should be discarded. That means that we now have a very strong indication that we can reconstruct the rise of the eukaryotes as a synergistic process in which two major forces, ATP generation very efficiently and internal enhanced ROS formation on the other side, were the two forces that really shaped the eukaryotes as we find them today. And that's indicated in this picture, in which you can see that you can almost find all the adaptations in the light of internal ROS formation. So this all seems like armchair science, you could say, especially in these conditions. But the interesting thing is that empirical findings now really show that symbiogenesis in a revolutionary period, in time scale you could say it's almost a restricted period, it really goes rather quickly. The eukaryotes evolve due to the two driving forces that I just mentioned. I hope I have convinced you that symbiogenesis is much better than all the more gradual models that seem to hold sway before the final research results over the last years, you could say, and that it's on the way out while symbiogenesis reigns supreme. There are a few publications that are really important, and these are listed at the end of this presentation. I hope you will check them out.